Is Dr. Fang here?
Yeah.
Okay, yeah, thank you. So he's going to give the next presentation on the human-induced pluripotent stem cell-derived corneal endothelial progenitor-like cell recovers the rabbit model of corneal endothelial dystrophy.
Hello everyone, I'm Fang from Chang'e Memorial Hospital in Taiwan. Today I'll show you my interesting study. The human iPSC-derived corneal endothelium progenitor-like cells recovers the rabbit model of the corneal endothelium dystrophy. I have no conflict of interest. We all know that the cornea transplant evolved from the PK in 1931 and followed by DSEC and DMAC in 2006. The first allogeneic corneal endothelium cell therapy was approved by Japan in 2023, and a similar clinical trial with magnetic allogeneic corneal endothelium cell therapy is also ongoing. However, young cornea grafts are necessary for this kind of therapy. Ideally, endothelium cells from human iPSC might be a better treatment strategy. This year, Professor Shimura published the first human iPSC-derived corneal endothelium treat Pullers keratopathy in humans. However, gene mutation of the injection cells was found despite the whole genome sequencing of the master cell bank. The development of the corneal endothelium was from the neuroclast and the corneal endothelium progenitor cell. And to address the previous issues, we think that maintaining a therapy cells in the corneal endothelium progenitor phase might be a good solution. We reprogram the patient's peripheral blood mononuclear cells into iPSC and then differentiate into the neuroclast cells and finally into the corneal endothelium progenitor-like cells, which we call IHC-PLC in this study. With the contact inhibition, IHC-PLC will become the human corneal endothelium-like cells. We intracamerally inject the IHC-PLC into the BAK corneal endothelium dystrophy model and reconstruct the mononuclear corneal endothelium. In the BAK-induced corneal edema rabbit model, BAK-treated cornea shows fibrosis, and the IHC-PLC injections group show the mononuclear corneal endothelium-like cells presenting with a low one and a human nuclear antigen, which means that these cells are from the human instead of the rabbit. External eye photography shows the corneal opacity in the BAK-treated eyes, especially in the postoperative week 1. As for the IHC-PLC injection group, the corneal edema resolved faster than the control. As for the central cornea thickness checked by the A-scan, BAK-treated only group were more than 1,200 micrometers, while the IHC-PLC group presenting gradually resolved the corneal edema. Now let's overview the result. We generated the neuroclast cell-like cell from the PBMC-derived human iPSC and differentiated into the corneal endothelium-like cells. And the differential gene expression between the IHC-PLC and the primary HCEC indicates a progenitor-like state of the IHCEC-like cells. And the IHC-PLC restore the cornea clarity in the rabbit's BAK-mediated endothelium dystrophy model. And the IHC-PLC displays a long-term sustainable therapeutic effect. The conclusion of this study is we customize the patient's corneal endothelium progenitor-like cells through the molecular induction. And the IHC-PLC have characteristics of the progenitor cells and is capable of proliferation in vitro. And the IHC-PLC provide sufficient therapeutic cells for preclinical efficacy and a safety evaluation as well as treatment. And this study was published in JAR. And thanks for Professor Chen, Professor May, and Dr. She's guidance and others team members help in this study. And this is my presentation. Thanks for your attention.
Thank you. We open it up for questions.
Yes, please. Yeah, sure. Can you show my PPT? I have the other data. As you can see in the IFC PLC RNA sequence, we can see the IFC PLC here, the second line is, and the next line is the primary HCEC RNA sequence data. We can see it's a little bit different. It's a little bit different, but it has the characteristic of the cell injection therapy like the CD44 negative, CD166 positive, and the CD133 positive and negative, and the CD24 negative and CD26 negative characteristic. And also it has the characteristic of the proliferation characteristic. And in the immunostaining of the IHCC and the B4G12, the corneal endothelium cell line, we can see actually this is the similar presentation of the low-1 and ATPA1A, CD56, CD160C, and Candarin, and the collagen AA1. And for the quantitative RT-PCR data, we can see the IHCC with the content inhibition, has the cell proliferation induction gene, and this left part is the IHCEC, and this is the B4G12 cell line, and we can see with the content inhibition, the proliferation activity will go down, and with the terminal differentiation gene will become increased. Currently, in the safety issue, we use the teratoma assay in the skin mouse, and there's compared with the initial IPS from the PBMC we inject into the skin mouse, and compared to the IFC PLC injection also in the skin mouse, there's no tumor found inside the mouse tissue.
While there is, if you go to the real-time data, from the eye, to go to the real-time data.
You mean what data?
No, the real-time PCR data.
Real-time PCR. This.
So the CD44 and the Wnt5b, if you see that there's a trend in the decrease.
Yeah.
But it's not. It's only less than a cycle difference, right? Or less than a fold difference.
Yes.
So there's a trend, but it's not really significantly more.
It's compared with the initial part. Yeah. Yeah.
So anything less than twofold, as you know, is only a cycle difference.
Yeah.
It's kind of dicey, but then we need to kind of look for more data.
Validate this.
Yeah.
With the content inhibition, they stop proliferation.
So what would be your next plan, having these results?
We are creating another kind of rabbit model, such as DSO, to check whether this kind of iPSC-derived cells can attach without the desmoid membrane.
So you'll be doing the safety and efficacy studies?
Currently, this data are only in the rabbit model.
Yeah.
Yeah.
Okay. All right. Thank you.
Okay, thank you.